So thank you for the opportunity to talk about this work that is basically aiming to move EEG and fNIRS out of the lab. So I'm going to present the work that we did on that in the last years. First of all, on the very left of this timeline, we see two brains from a book from 1879. It's sketches after the postmortem analysis of the brains of Gauss and the German farm worker. And comparing those anatomies, it was concluded that our brains work like a muscle. The more we use them, the more convolutions and mass they get. And I think it is pretty amazing to see that just within 1.5 centuries, we now have these powerful tools like fMRI, EEG, and fNIRS, and others that allow us to look into the functionality of the brain in vivo. Especially EEG and fNIRS have in recent years been miniaturized and made wearable, and so one of the potential new fields is to use them outside of conventional lab settings and to explore the human brain in more realistic and lifelike scenarios. This is of course also relevant for neurofeedback applications then. So in this talk, I want to focus on three main aspects of our work that are mainly modularity, open source of instrument, and the multimodal aspect in the signal processing, all focused on EEG and fNIRS out of the lab. And just recently, quite recently, there has been a very nice review on fiberless miniaturized functional near-infrared spectroscopy instruments that have been recently developed. And one of those reviewed systems is our open NIRS system that we published in 2015. That is NIRS only, but targets at providing very simple modular instrumentation for everybody to start with. That was our baseline, basically, system that we started to build on top on, and everything after is what I'm going to talk about now. The first is an instrument that is integrating EEG and fNIRS in a very miniaturized single instrument that is called M3VA. We then used that system for cognitive workload studies in freely moving subjects, and that's going to be more the signal processing aspect that I'm going to touch. And then I will give a very brief outlook on a new open source approach that we are currently working on at Boston University. So first of all, of course, there's a lot of challenges when we move out of the lab, and usually we try to tackle these by decrease the acquisition constraints, providing hardware that is actually enabling us to measure neuroimaging modalities. We want to increase the information that we can gather to build new signal processing methods that increase the robustness of our methods, and for that we need data sets that are multimodal and use them for the development of multimodal signal processing methods. And I will briefly go into details in one of our approaches there. So functional near-infrared spectroscopy, as probably everybody in this room knows, is not fMRI, is not as strong, but measures a similar biomarker, just limited to the cortex, and is based on also a very noisy signal, but based on near-infrared light. EEG, on the other hand, measures a complementary marker that is neuroelectrical activity. So both modalities are more or less complementary, but also in their spatial resolution and in their temporal resolution. So we have two modalities that are kind of enriching each other potentially. And there's more motivations why to use especially fNIRS and EEG for our applications out of the lab. So aside from the signals themselves that are complementary, the artifacts or the sensitivity to artifacts is as well. The additional physiological signals in these modalities are as well, can be used not only as artifacts but also used in our analysis. The instrumentation approaches are very similar in complexity and setup time, can be both made miniaturized and wearable, which is what is currently happening, are non-invasive and non-hazardous and comparatively low cost. So with these motivations in mind, we developed the M3VA as a hybrid EEG fNIRS architecture. The M3VA stands for Mobile Modular Multimodal Biosignal Acquisition. You can see one of those modules here. They are highly miniaturized and wireless and truly standalone, and they provide simultaneous EEG and fNIRS acquisition capability alongside with other biopotential measurements like ECG and accelerometer. We did extensive performance evaluation that I do not want to go into too much detail here now, but just briefly want to point out that the advantage of this approach, truly miniaturizing and putting it in a hybrid architecture, is that we have very low noise in both modalities and also minimized crosstalk between modalities in terms of noise. So these modules now allowed us to build custom-tailored headsets, basically, that is custom-tailored for the application that I want to talk about now. And so the background domain basically is brain-computer interfaces and neuroergonomics. So the idea here is to use neurophysiological markers as a support, basically, for a human operator in a feedback loop. And one of the main fields in neuroergonomics that are being investigated is the assessment of mental workload to adapt a system to the mental workload of the human operator. So there have been numerous studies, and also recently with multimodal studies with EEG and fNIRS. But however, that usually takes place in very controlled conditions, with the subject sitting at a table and is supposed to not move at all. And we wanted to basically go the other way. We want to go to this challenging environment where the subject is able to and has to move freely, but we still wanted very controlled conditions. And that is basically the novelty in this approach. To enable the acquisition of data, in the first place, we use two M3BA modules, put them in one custom-tailored headset that measures frontal functional infrared spectroscopy signals and frontoparietal EEG alongside with other signals. Just a very brief idea about the paradigm that we're talking of here. It is a modified spatial NBAC task in which a subject is standing in front of a wall with 8 buttons that are equidistantly placed on a circle, and the whole geometry is individualized to the height of the subject and also the wingspan. And during the experiment, the participant has to remember the position of a target color that is being told in the beginning of each block, and has subsequently to press buttons regarding a rule that is being told to the subject. So zero back in this case would mean always press the color of interest as soon as you see it, and 3 back means you have to remember 3 succeeding positions and then press the correct color. Now, as you could briefly see, there's a lot of movement involved, also bending down, and actually everything that we usually don't want in an experiment. The idea now is to exploit multimodality to tackle all the challenges that arise from artifacts to non-stationarities in the signals. Just briefly, the whole data set is 28 subjects and simultaneously acquired EEG, fNIRS, alongside with ECG and EMG in the neck, and movement of the head. So this is a very brief glimpse into the simultaneously acquired data with the M3BAs, and I want to briefly go into details regarding the functional infrared spectroscopy and acceleration signal. So here we can see what probably everybody would have suspected, and that is that movement is somehow modulating the signal, and that is very likely not the hemodynamic signal of interest, but physiological artifacts. So what we did then is focus on how to best remove those artifacts in an unsupervised way, but using multimodality. And what we see here is 2 raw intensity channels of functional infrared spectroscopy data. We have several bumps that are already a bit suspicious, so this data is bandpass filtered, but still, and they become even more suspicious when we add the button press events, which are corresponding to a certain body position or movement, and even more when we add the acceleration signals. We can see that there is some co-modulation going on. And basically, the method that we developed now is exploiting those co-modulations by performing temporal embedded canonical correlation analysis together with some other blind source separation approaches using a ICA that exploits higher order statistics in the data. And this framework, as a new analysis framework for fNIRS, we called Blizzard. Because of the time reasons, I don't want to go too much into detail, but just briefly show basically before and after signals. Here is just one channel, oxy and deoxyhemoglobin, during 30 seconds of the experiment, and you can see that the signals, the variability of the signal due to movement is heavily reduced. This work was recently also accepted for publication, so I hope soon people can read about it. But we did also extensive evaluation using ground truth resting state data augmented with simulated HRFs, and performed compared performance with other methods. And here in the last picture we see before and after removal average artifacts, so basically just the signal in two channels for each different motion position or body position, averaged across all trials and subjects. And so after this method has been applied, we basically are rid of these average artifacts. So using that method, now we use that method to clean the fNIRS signals with the accelerometer signals. This pipeline is very simplified and shows only some of the main aspects, but on the fNIRS side, we then look into typical hemodynamic features, maximum and average in a classification setting. And on the EEG side, we regressed out EOG signals and performed multi-band common spatial pattern analysis for alpha and theta bands, commonly known to be modulated by workload, and perform a hybrid classification across validated regularized linear discriminant analysis to get some classification of workload stages. And again, this is a little excerpt only of the results, but the main important message is that we were able to discriminate different levels of workload in different time windows down to 30 seconds with more than 80% accuracy despite heavy artifact rejection, and that these classification accuracies are in the same area, in the same realm, comparable with results reported for other NBAC-based fNIRS EEG studies in sitting subjects. And so we found this very encouraging to move on with this technology in, let's say, challenging conditions outside of the lab. So I want to briefly now use the remaining minutes to give a very brief outlook on the work that we're currently doing at Boston University. As I briefly mentioned, the first generation open source functional infrared spectroscopy device is a very simple device that can be used for anybody who wants to develop their own technology. Basically, it's a good starting point, but it could not be used for whole head diffuse optical tomography imaging. So this device is much more sophisticated, potentially also more expensive, but also open source, and is truly scalable, can be used with 2 to 128 optodes, and will be extended to EEG and NIRS very soon. So this system is going to be available hopefully by the end of the year, at least in terms of the production files. And yeah, that's what we are working on right now at Boston University. One of the ideas that we are also following with that right now is using the established HOMER and Atlas VIIR framework for a functional infrared spectroscopy analysis that that has been used so far also to create caps and patches to place fNIRS optodes depending on the region of interest in the brain. And now we're actually currently working on automizing this approach, where you can design your EEG and fNIRS cap based on the regions of interest, and then this cap is being printed with the holders exactly where they should be, basically. So lastly, just briefly pointing out again these two resources, opennirs.org for a very simple beginning, and openfnirs.org for the much more complicated and extensive system that is going to be available soon. And I'm actually a bit early, so maybe we have time for questions, and I thank you for your attention.